All right. Well, good afternoon, everybody, and welcome to this, the first Dean's Lecture for 2019. As you know, earning the rank of professor in the Bloomberg School of Public Health represents an incredible achievement and confirms not only the respect and high regard of one's peers, but also of the school and the faculty in the school and the colleagues in the School of Public Health. The Dean's Lecture Series was designed to introduce the Hopkins community to the work of newly appointed and promoted professors, and also to provide a forum to promote the interchange of interdepartmental awareness and collaboration. We are looking forward to a terrific series for 2019, celebrating just some of the faculty that really make this school great. So to kick this series off this year, we are very pleased to have with us today Dr. Anna Durbin. Dr. Durbin studies the human responses to live attenuated vaccines with a primary focus on dengue. At the Center for Immunization Research, she has conducted numerous phase 1 trials, vaccine trials, with the ultimate goal of identifying suitable monovalent vaccine viruses for inclusion in a tetravalent dengue vaccine. She is particularly interested in studying the immunopathogenesis of dengue infection and disease. And just last year, Dr. Durbin received $1.8 million in NIH funding from the NIH to evaluate a new Zika virus vaccine. The study will evaluate the safety and immunogenicity of the vaccine in adults with no history of previous infection. And you'll hear a lot about all of that work in just a minute. But Dr. Durbin's work has resulted in over 100 publications in peer-reviewed journals and a couple of book chapters, and she has given many invited presentations at scientific meetings and invited lectureships. She has served on national and international advisory boards and subcommittees committees related to both dengue and malaria vaccine safety. So Dr. Durbin came to the School of Public Health in 1999 as a young assistant professor in international health in the Center for Immunization Research. She was promoted to associate professor in 2007 and then to full professor in 2016. She also holds joint appointment in the Department of Medicine in the Division of Infectious Diseases. And she's an attending physician in the Moore Clinic, the HIV outpatient clinic of the medical institutions. She is a terrific advisor to many students in the department and co-instructs the course Biological Basis of Vaccine Development. She also directs the Vaccine Science and Policy Certificate Program in the Department of International Health and is a valuable member and a longstanding member of our Institutional Review Board. Dr. Durbin received her BS in pharmacy from the University of Michigan and received her medical degree from Wayne State University, where she stayed on to complete her training in internal medicine and infectious diseases. Following her medical training, she was a clinical fellow in the respiratory virus section of the Laboratory of Infectious Diseases at the National Institute of Allergy and Infectious Diseases at NIH. She has received numerous awards, and let me just name a few. She received the NIH Merit Award for Outstanding Basic and Translational Research in Developing Vaccines for the Prevention of Respiratory Virus and Flavivirus Diseases. She received the Instituto Butantan, if I've got that right, medal from the government of Sao Paulo, Brazil, and for the dengue vaccine development, the NIH Director's Award, and the Vaccine Industry Excellence Award for Best Academic Research Team. I will say two things. First of all, Dr. David Peters regrets that he cannot be here. He is in Russia somewhere, but he sends his congratulations and greetings to everybody. And I will also say that in meeting with Anna earlier this week, I was truly swept away by her enthusiasm for the work she does, and also left very optimistic about the future of vaccine trials, given all the great work she's doing. So please join me in welcoming Professor Anna Durbin.
Well, it's really, truly a privilege and an honor to be here, and I think thank Dean McKenzie for that wonderful introduction. How do we stuff 20 years of work into a 45-minute suitcase? Well, I'm not going to talk about all of the things, of course, that I've done over the past 20 years, but I do want to highlight some things that I think are very important, and I really want to emphasize that it is a team effort. I see a lot of my team members here from the Center for Immunization Research, but it's teams within Hopkins and within the center, within the school, within the university, and then, of course, outside the university that really helps us do the science that we do. I do want to give just a little bit of background on dengue, just to sort of orient you a little bit to the importance of vaccine development and where we are with vaccine development and what some of the, I would say, hurdles are to vaccine development for dengue. So dengue is really endemic in all tropical and subtropical regions of the world. When we talk about a dengue vaccine, we're really talking about 4 individual vaccines because there's 4 dengue virus serotypes, all of which can cause the full spectrum of disease. And we know that infection with one dengue serotype may confer lifelong protection against symptomatic infection with that particular serotype. It only confers very short-term protection against the other 3 serotypes, and in fact, may actually predispose to more severe infection down the road later on when people are infected with a second different serotype. We know that secondary infection with a heterotypic or different serotype is strongly associated with severe disease. We think that that's due to antibody-mediated enhancement of infection, which I've just sort of illustrated here, but which you don't really need to know too much about for this talk. The bottom line is what we sort of summarize it as is partial immunity to dengue is bad. And that's something that you want to remember throughout, if you're listening or talking about dengue or hearing about dengue or dengue vaccines, just remember partial immunity to dengue is bad. And I'll show you an example of that in a little bit. So first of all, what is a dengue controlled human infection model? Because I will say when we first started to work on development of this, we got a lot of jaws dropping open and why are you doing this and what is it and how can you do that? And when I talk about it and when I was writing this slide, I looked at that and said, some people might raise their eyebrows when I say that normal, healthy volunteers are infected with the dengue virus and then we follow them for signs and symptoms of disease. How can we do that? How can we do that in an ethical manner? And I'll talk a little bit about that. The concept is really similar to the controlled human infection model, which has become a very, very useful tool for the down selection of candidate malaria vaccines and really has become an integral part of that evaluation process. Most malaria vaccines today will not move forward without an initial evaluation in the controlled human infection model for malaria. The difference between dengue and malaria, however, is that with malaria, we can give volunteers medication that will treat them before they become symptomatic with malaria illness. We don't have such a medication for dengue, which is not to say we can't treat dengue. We can treat dengue symptomatically for people who would develop symptomatic illness or severe disease. Our goal, however, is not to allow that to happen. Our goal is to have an infection model, not a disease model, and I'll touch a little bit more on that. So why did we elect to develop a dengue controlled human infection model? I will say that in the past 20 years that I've been working on dengue, we've had sort of a paradigm shift when it comes to dengue vaccine development. We know, as I said earlier, that partial immunity to dengue is bad and that a second different dengue infection can cause more severe illness. And we were always concerned working with live attenuated dengue vaccines that if we took a live attenuated dengue vaccine into a dengue endemic area, that vaccine might make people ill because of antibody-dependent enhancement of infection. So people who already had dengue antibodies, a live vaccine would replicate to higher titer and may cause more severe disease. I will tell you that that paradigm has been turned upside down, and we now know that the people who may be at greatest risk from a dengue vaccine are people who have never had dengue before, and I'll show you some data around that. But it started with the phase 2b efficacy trial of the dengue vaccine CYD, which is now called Denvaxia. We had thought prior to this trial that neutralizing antibody to dengue was a correlate of protection. all you needed to do with the dengue vaccine is induce neutralizing antibody, and people will be protected from subsequent dengue infection. We had an incomplete understanding of the immunopathogenic mechanism of dengue, however. We knew that antibody-dependent enhancement of infection was a player in it, but there were also concerns about the T cell response and cross-reactive T cells that may be contributing to dengue. And so we thought maybe we can use a dengue-controlled human infection model to really try to dissect out those immunoprotective versus immunopathologic responses following vaccination. And we also, after looking at the phase 2b and phase 3 trial results, really wanted to ensure that any candidate vaccine that moved into endemic areas, that we had evaluated it as much as possible to ensure that it really did have the best chance of being successful and not inducing an immunopathological effect, which I'll talk about with Denvaxia or CYD. What we wanted to know is, can we find a better predictor of vaccine efficacy other than neutralizing antibody, because neutralizing antibody failed as a very good predictor. And these are the data to show that. So this is from the phase 2b efficacy trial of CYD, which is now known as Denvaxia. These are the antibody titers, and this is the percentage of seropositive volunteers following vaccination. So we see here that 99% of volunteers who were given Denvaxia developed neutralizing antibody to dengue 2. When they looked down at the efficacy results, however, it completely failed to protect people against dengue 2 infection. So our sort of gold standard, what we're looking for in terms of a vaccine to predict protection, failed. And then when we moved on to the phase 3 trial of this vaccine, this was CYD14, which was conducted in 10,000 kids in Asia of Denvaxia. What they found is in year 3 of the trial, so the first year is given to vaccination. The vaccine is given as 3 doses over the course of a year. The second year was the primary efficacy period, and the third year was the long-term safety assessment. What they saw is that those kids who were 2 to 5 at the time of vaccination had an increased relative risk of hospitalization in year 3 if they received vaccine compared with placebo. The vaccine predisposed them to hospitalization for dengue. So that's not what you're looking for in a vaccine, certainly. Now, one of the questions that we had there, was this an age-related phenomenon? Because you can see that relative risk goes down with age, or is it something else? And unfortunately, in the phase 3 trial, there weren't enough samples collected to really look at that. What I will say is that this vaccine is now licensed only for kids 9 and older up to adults age 45. And it's only licensed for people who have already had dengue, because subsequent studies showed that the people who had never had dengue before were at higher risk of this happening, as opposed to people who had already had at least one episode of dengue before. So the vaccine is currently licensed only for people who have already had dengue and are 9 to 45 years of age. So this comes back to why I think we need a dengue-controlled human infection model. We know now that neutralizing antibody in and of itself is not a good correlate of protection and not a good predictor for vaccine success. We also wanted to investigate the role of cellular immunity, and I won't go into great detail, but one of the characteristics of the CYD vaccine is that it does not contain CD8 epitopes of dengue. It has CD8 epitopes of yellow fever because it's a chimera. But again, we know that a dengue vaccine that fails to protect may not just fail to reduce disease, but may actually result in increased incidence of disease if it only induces partial immunity. So we wanted to minimize the risk as much as possible of introducing an ineffective or a poorly effective dengue vaccine into endemic areas. And to do that, we thought that a controlled human infection model will help us choose the best candidates to move forward for further testing in endemic areas. So then we had to come up with what we felt were the ideal characteristics of a controlled human infection model. And I'm not going to go into a lot of discussion about this, but we wanted an infection model as opposed to a disease model. So our goal was not to induce symptomatic dengue fever, or severe dengue. We thought that the risk of that was too great. What we wanted to do was was develop an infection model. And part of that comes back to what we know about the natural history of dengue. So about 3/4 of dengue infections never present for clinical care. So they may have a low-grade fever, they may have a rash, but they never present for clinical care. Only about 25% of infections do. And for that reason, we felt, one, if we tried to push the model toward a disease model, we're going to make people very, very sick. And two, we felt that we could then create a good model just looking at infection. And by infection, what we were looking for was evidence of viral replication. We do typical tissue culture, but I'll show you data also where we've looked at it by PCR, and then to see if there were any clinical signs or symptoms as well. We wanted endpoints that would not put the volunteer at risk for severe disease. So we, as I said, did not want to cause dengue fever where people would not be able to go to work or go to school or be incapacitated. We certainly did not want any evidence of dengue hemorrhagic fever. And so we felt that we could do this if we chose the right strain for dengue virus and if we chose the right dose and if we followed them closely. So we actually started with a failed vaccine candidate virus. The failed vaccine candidate virus is dengue 2 Tonga 74. It was originally developed as a vaccine candidate because it was described as being naturally attenuated in epidemiological studies of a dengue 2 outbreak in the Kingdom of Tonga. So it had been described as causing milder disease, lower viremia. So we thought it was a good candidate to start looking at as a vaccine candidate strain. And 30 nucleotide deletion was introduced to further attenuate it for vaccine purposes. However, when this virus was studied in a non-human primate model, it did not appear at all attenuated compared with the wild-type parent virus. So it had the same amount of viremia in the non-human primate. So we put that on the shelf and said, we're not going to use this for a vaccine candidate. But we then brought it back out and repurposed it for use in a challenge study. It was made under good manufacturing processes, was vialed up, and we took it into the clinic to evaluate it as a challenge strain. Excuse me. We started by enrolling a very small study, only about 14 subjects. 10 got the challenge strain, 4 got placebo, just so that we could compare any of the adverse events and make sure that they were truly related to the challenge strain. What we found is that we were able to recover replicating virus from 100%, all 10 of the people who received the challenge virus. The levels of viremia were about 100-fold higher than we'd seen with any of our vaccine candidate viruses. And we also were able to detect some clinical signs, particularly rash. So we saw a rash in about 80% of subjects who received the challenge virus in this study, and neutropenia in about 20%. The neutropenia was very short-lived and did not did not put the volunteers at any risk for any subsequent bacterial infection. But importantly, no subject developed fever, no subject developed LFT or liver function abnormalities or any signs or symptoms of vascular leak. They all went to work. They didn't miss any work. They didn't miss any school. This was all done on an outpatient basis where they felt pretty good, just noticed the rash. And this is just to show you the rash. It is quite noticeable. It's total body rash generally, head to toe, and it itches just a little bit, although interestingly, only about half the people actually felt it itched enough to take Benadryl or any medication for that. Most of them didn't take anything, and those who did take something only took a dose. This is the challenge virus rash here, and this is compared with the rash that we typically see with our live attenuated dengue vaccines. It's very, very different, but very easily noticeable. So this is just a list of the challenge studies that we've subsequently done with this virus, but I'm only going to highlight 2, and the 2 studies that I'm going to highlight are ones that I picked up because I felt that they really advanced our studies of vaccines, and they've also informed us in terms of looking at immunoprotection or immunopathology for dengue, and really helped us move forward in what we know about vaccines and vaccine development for dengue. So this study was our first vaccination followed by challenge study with dengue 2. This is our live attenuated tetravalent vaccine here. It was called Formulation 3. I will say we performed about 18 clinical trials just to come up with the viruses that would be included in TB005. And TB003 and TB005 are our two lead tetravalent candidates following all of the clinical trials. We felt that TB003 was a weaker candidate in terms of its immunogenicity against dengue 2, which is why we chose this as a stringent challenge study. I will say that the dengue 2 component in TB003 is a chimeric virus on our dengue 4 component, and the parent strain of the vaccine is a different strain than the challenge strain. So it is a heterotypic strain, not a heterotypic serotype. They're both dengue 2. But we brought in 48 subjects, 24 received vaccine at day 0, 24 received placebo. And then 6 months later, we brought those people back, those people who were eligible and for challenge were challenged. We had 42 people who were challenged. About half were TB003 recipients, half were placebo recipients. And then our primary efficacy endpoint was protection against viremia. Our secondary endpoints were protection against rash and neutropenia. We were able to power this study very easily with just 48 people because of the high incidence of viremia that's induced by the challenge virus in placebo recipients. Because 100% of them were viremic, we can power these studies with relatively few subjects. So this was a single dose of TB003. This is the antibody response. The only thing I want to point out here is that we had 100% with neutralizing antibody to dengue 2, and we had 100% with the trivalent or better response. So people responded very well to this, to a single dose of TB003. When we challenged, this was borne out in our challenge data. So we had in our placebo recipients, 100% again, had detectable viremia by culture. None of the TB003 recipients who were challenged at 6 months could we detect any virus. We looked by culture, we looked by PCR, we looked by digital drop PCR, and we could find no evidence of viral infection when we looked at viremia or even when we looked at different clinical outcomes. So again, we had 80% of placebo recipients develop a rash post-challenge, 20% develop neutropenia post-challenge, but none of the TB003 recipients developed any of that post-challenge. The only notable difference, I will say, is when we're looking for infection, one of the things that we look for is a boost in antibody titer, because the thought is the virus has to get in, has to stimulate the immune system before you'll see a boost in antibody titer. So of the 21 vaccine recipients who were challenged 6 months later, 12 had no boost in antibody titer, as defined as a 4-fold or greater rise in neutralizing antibody response to dengue 2, but 9 did. And when we looked at their titer to dengue 2 prior to challenge, just prior on day 180, the titer in those people who did not have a boost was significantly higher than those who did have a boost. So this was our only indication that there may be something different in these two groups of people. And I bring that up because we're going to try to use this study and look for correlates of protection. So what did the dengue 2 challenge study told us? It said, yay, the vaccine looks good. You can protect against dengue 2. You protected everyone against dengue 2 infection. That's great. The problem is when you're looking for a correlate of protection, you really need some people to fail, and nobody failed. So that's why we were looking at every little thing, and that's why I mentioned the 4-fold rise in antibody titer or greater. But we did still, and I'll show you some data, we still did go back to the study and really analyze specimens, trying to find indicators of viral replication post-challenge. The good news is, and Dean McKenzie mentioned the Butantan medal, it's really been my great pleasure to work with the people from the Institute Butantan in Sao Paulo, Brazil for almost 10 years now. They were the first group to license the NIH dengue vaccine. They licensed it when there was only a dengue 4 candidate. They had faith that we were gonna go and get other candidates. They started their phase 3 trial about 3 years ago. They've enrolled 17,000 people in their phase 3 trial. This is their artwork to describe. You can tell this is the vaccine candidate because it's attenuated. They have bandages. And this is the chimeric component. Good for Brazil. There hasn't been a lot of dengue in Brazil the past 3 years. Bad for the vaccine trial. We're hearing that there's more dengue in Brazil. We're cautiously optimistic we may meet some efficacy endpoints by the end of this dengue season. So keep your fingers crossed. It is a 5-year safety follow-up, and that goes back to what was seen with the Sanofi Pasteur Denvaxia study. We need long-term safety follow-up to make sure that the vaccine does not induce more severe disease compared with placebo recipients. So can we use the CHIM to try to look for correlates of protection? We think that the CHIM is an ideal sort of process to do that because you can control everything. That's what the C is for, controlled. So you can control what they get, when they get it. You know exactly what their immunological background is in terms of flaviviruses before you challenge them or vaccinate them. The viruses are well characterized. We know the interval between vaccination and challenge. We collect a lot of blood over many frequent time points. And we collect blood for all different kinds of analyses. And we think that that really is a wonderful way to try to study this. I will say no one person can do this, right? So it's also a great opportunity for collaborations. And I made some really great collaborations in trying to do this work. And we're going to see some of that in just a little bit. So for the correlates analysis, we took all of the clinical data. We took some neutralizing antibody assays and other antibody assays that we developed for this purpose. We took the B cell assays, the T cell assays, and we took all of that data and we compared data points post-vaccination in people who received vaccine, post-challenge and placebo recipients who were challenged, and then tried to compare that and see if we could pick up some indication of viral replication in those TB003 vaccinees who had been challenged. Then those different variables were mapped into a network to try to see how they relate to one another and to look again and see if we're seeing the same kind of mapping post-challenge as post-vaccination and try to find some evidence of viral replication. And there are a lot of people I'll show you at the end who did all of this work, the people who are collaborators in Vermont who were working with doing T-cell analyses. We did antibody repertoire mapping, post-vaccination and post-challenge. Dr. Andrea Cox from the School of Medicine and I'm going to show some of the work that she's done, has done some really interesting work as well. And we're trying to do all of this and integrate all of it and try to get some indication of what might relate to protection, what might relate to immunopathogenesis. So these are all of the different variables. You don't have to read that. I don't expect you to read that at all. Just to sort of walk you through it, these are the days post-vaccination or post-challenge. We're looking at post-vaccination now, so this is day 2, 4, 6. These are some of the time points. And these are the unadjusted p-value, the log 10 of the unadjusted p-value. So anything that's red, nothing significant. Anything that's orange, not really significant. So when you have all of these different variables, how do you even see if there's significance or not? Well, this is the unadjusted. When you adjust it, you see a lot falls out. No surprise there. And what we're really looking at, what's the difference between the day of vaccination and post-vaccination? Well, you get an antibody response, which is this over here. You see some changes in white cells, lymphocytes, monocytes, et cetera. You see some CD4 changes, some CD8 changes, and that's really all of what that comes down to. Now, when you look at that post-challenge, so these are the vaccinees who were then later challenged, really nothing, right? So again, what we're seeing is very little evidence that there was any viral replication post-challenge. This goes back to supporting that the vaccine was very, very protective, which we like to see. This is the placebo recipients who were challenged. And again, you see something, a little bit more indication of what might be called viral replication in terms of, here's antibody responses again, and then we have our white cell counts. We have different things that change post-vaccination in the different assays that we looked at. But the point is it's very different between post-vaccination if you receive, post-challenge if you receive vaccine, and then post-challenge if you receive placebo. So this is the network measurement that was put together. And just to sort of how you interpret this, and this is data that was just presented a couple weeks ago on one of our conference calls. And it looks really complicated, but it's not that complicated. And really what this is trying to tell us is, are things related or not? How do we look at them in relation to one another? Are some responses more related to others? I think that it's a really nice way to look at things, but I think there's a lot of caveats that go with this. And this is really preliminary representation. But what I wanted to show you is we're looking at vaccinees post-vaccine. So what do we see? We see dengue 1 antibody responses here, and each serotype is sort of coded in blue. Dengue 2 antibody responses here, dengue 3 antibody responses here, dengue 4 antibody responses here. What we see is the CD4 response is linked or correlated with the antibody responses. So that's not really a big surprise there. We have two different colors for dengue 2 because we included both the challenge virus and the parent virus in the analysis. And I only put this up here because we're going to look at this post-vaccination and then compare that with post-challenge. So again, post-vaccination, just to highlight some of this is our dengue 2 and our dengue 3. Those antibody responses appear to correlate more with one another. What does that mean? The titers appeared to correlate. They were closely related. Dengue 1, the titers, I will tell you, were lower. They didn't correlate quite so well. And then dengue 4 was off by itself a little bit. But we did see responses, CD4 responses to each of the 4 serotypes in this. The CD8 responses here post-vaccination are included, not post-challenge. That analysis is still going on. So then when we look at post-challenge, what do we see? This is, and I didn't blow this up, but what you see is a big response to dengue 2. That's not a surprise. They only got dengue 2. So we don't see those responses to the other 3 serotypes, right? We only see the response to dengue 2. We do see the CD4 and CD8 T cell responses induced by the challenge virus in those who received placebo. We see much smaller responses here in the TB003 recipients. So what we see again is that there appears to be a lower response in the vaccine recipients, particularly when we compare that with those people who got placebo first. So the only, right now we're still doing this analysis, and some of, as I said, the CD8 responses post-challenge are still ongoing. What was interesting, what I thought was interesting, we do all this work, it's taken weeks to do all of the statistical analysis and then determine what's significant, what's not significant, do our network analyses. And what do we see? We see that, the only variable that appears to stand out is a boosting antibody titer to dengue 2, which we saw that early on. We were hoping we would be able to correlate something with that. So far we haven't, but we might not be done yet. And that's where I'm going to get to our trivalent and some of the exciting data that we have with that. So because we had such good protection and we're really unable to tease out a good correlative protection from the vaccination followed by challenge study, we said, can we push our model to failure? Can we push our vaccine to failure? Because we need some people to fail to be protected if we want to find a correlative protection. So we decided to make a vaccine with 3 of the 4 components from the TB003 vaccine. So we took the dengue 1 component, the dengue 3 component, and the dengue 4 component, and we administered it to volunteers, and we had some placebo recipients in as controls. And then 6 months later, we challenged everyone with dengue 2. So these people had never seen dengue 2. They'd never seen any dengue 2 antigen, and we challenged them with dengue 2 to see if they were going to be protected. This is the, I just put this up here so that you can see the only difference, the only antigenic difference between the trivalent component and the tetravalent component are these antigens, the preem and the envelope. And it's thought that the envelope protein is the one that confers protection. So we have the same non-structural proteins. We think that CD8 epitopes, 98% of the CD8 epitopes to dengue reside in the non-structural proteins. The non-structural proteins are exactly the same. The only antigenic difference is the presence of really the E antigen. So our question was, will this trivalent formulation protect or won't it? We know from natural epidemiological studies that generally your 3rd or 4th dengue infection does not lead to hospitalization. You don't present for care. They appear to be much less symptomatic than a secondary dengue infection. And the dogma has always been that trivalent should be fine. You should be able to protect with 3 of the 4 serotypes. You don't necessarily need a tetravalent vaccine, and that's what we wanted to test in this study. So I said these people have never seen dengue 2 before, yet we still had 22% of them develop neutralizing antibody to dengue 2. We know that neutralizing antibody is cross-reactive. It was not generated by dengue 2. It was generated by dengue 1, 3, or 4. So what's interesting is those people who, we had 21 return for challenge, 15 of them had received the trivalent, 6 had received placebo. Some of those, as I said earlier, had had dengue 2 antibody detected at some point prior to challenge. What's interesting is, so we go back to our tetravalent where we had nobody who we could detect virus by culture PCR or digital drop PCR. In this, we had 4 out of 15 recipients who we could detect dengue 2 virus by culture. And most would say, well, that's pretty good. You were able to protect 11 out of the 15. That's pretty good. But when we looked by PCR, we were able to detect virus in 12 of the 15, which is a stark difference from what we saw with our tetravalent recipients. So if you look here, we had trivalent recipients, controls. Here we had 4 we could detect virus by culture, 12 by PCR. I will say that the titer in those people who had breakthrough viremia was no different from the placebo. So if you broke through, you broke through. You had a very good titer. It did not appear to be abrogated at all if you had received the trivalent. So then we wanted to go on and say, is there anything else we can do? And I was just having a conversation with Dr. Andrea Cox, and she's like, I work on hepatitis C virus, and we've really looked at IL-18, and we've been able to detect IL-18 rise very early in viremia. Did you ever think of looking at IL-18 as a marker of viremia? And I said, no, because I don't work in IL-18. And she said, well, we'd love to look at that. Can we collaborate? And I said, that would be absolutely fantastic. And so this is the work by Laura Cohen and Andrea Cox from Andrea's lab. And I just want to walk you through this really quickly because these data are relatively new. And I think it's giving us something that we may be able to use at least in future studies and go back and reanalyze some of the work that was done earlier. So this is looking at IL-18 post-vaccination. The blue line is the TB003 recipients. The pinkish line here, salmon line, is placebo recipients, and we see a nice rise in IL-18 post-vaccination in just about every vaccinee. So even, I will say, post-TB003 administration, we're only able to recover vaccine virus in about 60% of vaccinees, not in everyone. If you look at post-challenge, what I was very intrigued with originally is we get a higher peak here in the challenge virus. So these are, again, the placebo recipients who were challenged with dengue 2. And even though it's a low dose of dengue 2, it's 1,000 virus particles. When we look at the total dose of vaccine that's administered, it's about 4,000 virus particles. We still see a higher peak. The other thing that we see in some of these vaccine recipients is we actually do see a rise in IL-18. And whether or not that is significant and correlated with a boost in antibody titer, we don't know yet. We've not gone back and looked at that, but that's something that we want to do. And what I thought was very interesting is if we now look at what happens post-trivalent administration in challenge, this is what we see. And this is broken down into 3 different graphs here. What you're looking at, red is those people who were not vaccinated, essentially placebo recipients. Those people who were vaccinated and did not have vaccine virus recovered, and then those persons who were vaccinated and did have vaccine virus recovered. So again, in the vaccinees, we see a nice boost in IL-18 post-vaccination. Post-trivalent, challenge of those people who received the trivalent vaccine or placebo. What's very interesting is we really see no difference between the people who received the trivalent and were challenged and were viremic post-challenge and those placebo recipients who were challenged. And not only that, if you look at the peaks of IL-18, And I think this is very, very interesting. It's higher in the people who received the trivalent and then were challenged compared with placebo recipients who were challenged. And Andrea and Laura are looking at that now and looking at the role of the inflammasome and whether or not this has to do with antibody-dependent enhancement of infection or other mechanisms. But it's very, very interesting that in these people who received a trivalent vaccination, they actually had, to some extent, greater IL-18 production than even the placebo recipients who were then challenged. So whether or not, again, this is an indicator, could this be a marker of risk? We don't know. It's something that we need to look at in additional studies, but I think does give us an additional tool to go back and look at our correlates analysis and try to incorporate this and see if we may be able to find some evidence of viral replication that can help us better understand what we really need for vaccine protection. I do think we learned a huge lesson with our trivalent followed by challenge. And what this study told me more than anything else is that you need homotypic antibody if you're going to prevent infection. Now, you may not need to prevent infection to prevent disease. That's a really high bar. But we do know, at least with dengue from this study, that if you do not have homotypic antibody, you are unlikely to prevent infection. Now, in those people who received the trivalent vaccine and were challenged, as I said, only 4 of them had virus that we could recover by tissue culture. We had 8 others who had virus recovered by PCR. What that tells me is that they were able to control their infection before it reached a level where we could actually find infectious replicating virus. So whether that's the cellular immune response that's able to contain that infection, those are studies that we're doing now. But it does tell me that if I want to prevent infection with the dengue virus, I need homotypic antibody. That heterotypic antibody is just not enough to do it. And we still think that, again, may actually be predisposing to disease. We also learned from this that IL-18 is a very sensitive indicator of viral replication. And again, that may be something that we can add to the toolbox to go back and look for evidence of viral replication and add that to our correlates analysis. So in summary, I just want to say that I think these studies have been just tremendous, just very, very rewarding, not only from the science point of view and what we can bring and to the understanding of dengue, but also to the collaborations that I've been able to establish over the years, because really that's what's fun about science. It's meeting different people and talking to them and coming up with ideas and trying to broaden what we can learn. And certainly, I think the dengue controlled human infection models are a great tool, but you need a really great team, or you need a team of teams in order to really do the work and fully, I think, realize the potential of these models. The CHIM was really key to the Institute Butantan choosing TB003 to move forward for their phase 3, and I think that was very rewarding. But I think the studies that we can do and the samples that we collect and the design of these trials, we can design CHIM trials to answer specific questions about a vaccine, a therapeutic, or even just about dengue immunity and immunopathology. And I think that meeting people who can help with that has been just incredibly rewarding and makes it all worthwhile. The other thing is that the clinical trials themselves, which is really what my role is designing and conducting the clinical trials, needs so many really, really excellent people to make that work. The team that we have at the CIR, I think, is really unparalleled, and that was exemplified by the award that was given for Best Research Group, Clinical Research Group. The work that they do is really outstanding, and these trials can't be done. There's so many people on so many levels that need to really do their job and do their job well to have these trials done. It's been really a great pleasure and honor to work with everyone at the CIR. I think there's a lot of work that we can do in the future. I haven't talked about this, but we've developed a dengue 3 CHIM model as well that we're starting to use, and we've looked at to look for vaccine efficacy. And we're hoping to start a Zika-controlled human infection model trial probably after September of this year, if all goes well. And I think that's going to be very valuable in understanding the interactions between dengue and Zika or other flaviviruses. So we're very excited about this. This is just a picture of our original CHIM consortium that was funded by the Gates Foundation and the NIH. I will say, though, it's broad, and this is, I wish I could put everybody's name on this slide because so many people contributed to the work that I presented. But I'm really proud of the folks at the CIR and all of our collaborators and the work that we're able to do. And I'm very excited about new CHIM studies that we're going to be doing in the future. Thank you.
So we do have some time for questions.
Yes.
We have some microphones.
Hey, Dee.
Great, Anna. I wanted, you excluded, should I go to?
Yeah, here, I guess. Yeah, that's great.
I think it's. You excluded individuals with prior flavivirus infection from the challenge studies, but I wonder if you could just talk a little bit about the potential impact of exposure to other flaviviruses on the dengue vaccine.
That is a great question. So we do know that other flaviviruses do induce cross-reactive antibody to dengue. This was a big concern, particularly with Zika during the Zika outbreak, and whether or not pre-existing dengue antibody influenced the incidence of the congenital Zika syndrome. And that is something that we're actually hoping to study with controlled human infection models in two different ways. One, there's a grant from the Wellcome Trust that we're working with Bridget Wills and her group in Vietnam to actually bring the dengue CHIM to endemic areas and look specifically at people who have prior immunity to dengue and perhaps other flaviviruses. The other studies that we want to do is bring people back who, when we screen people, we exclude people who have evidence of prior infection, but we can design a study to look specifically at those people and challenge those people. The study that I really want to do is give people, enroll people and give them the dengue challenge virus, and then down the road give them Zika and vice versa and see what the effects on that is. And that's really the study that I'd love to do after we get our Zika controlled human infection model established. Yeah.
Other questions? Yes.
Thanks, Ellen. Anna, that was fantastic. Can you say a little bit about what you think the implications of the Denvaxia trial in Southeast Asia will be on dengue vaccine research broadly and the use of vaccine in the future?
No, I think that's a great question. And I have to give Sanofi and Denvaxia a lot of credit. They really worked very hard to try to understand what's going on. It's really changed our whole perception of dengue vaccine and what we need to induce protection, how long we follow people, what we say to people about risks of dengue. I think, and I didn't have time to go into it today, but I will say, I think the dengue vaccine, CYD, has a lot of flaws, and I think there were certainly evident red flags early on in clinical development that because we had this understanding or we thought that neutralizing antibody was enough, that we didn't really pay attention to them. And I had said at the end of the CIR 300 study that homotypic antibody we think is very important for protection. What we know from Denvaxia and from the studies that were done subsequent to the phase 3 trial is that it induces great antibody, homotypic antibody to dengue 4. The dengue 1, 2, and 3 components are not very infectious, and likely the antibody that we see to dengue 1, 2, and 3 induced by Denvaxia is not homotypic, it's heterotypic. It's cross-reactive and induced by that dengue 4 serotype. So what that told us is we have to ensure that every component of the vaccine is infectious, and we have to be able to show that it's not cross-reactive. And a collaborator, Dr. Aravinda De Silva, has developed an assay to look at, he essentially depletes the serum of antibody to 1, 2, 3, or 4, and then measures it again to see if that antibody was cross-reactive or whether it was indeed homotypic. And we've developed an assay we call the neutralization capacity assay, because I think one of the things when we use the NEUT assay, and I'm not going to go into how we do it, but I'll just say that it doesn't measure non-neutralizing antibody. It only measures neutralizing antibody. And we think that non-neutralizing antibody is really what's responsible for antibody-mediated enhancement of infection. So it's very important, and you can look and you can be seeing in your NEUT assay, this much neutralizing antibody, and you might have this much non-neutralizing antibody and you won't know it. So I think it's really taught us to think much more carefully about how we evaluate vaccines. The second live attenuated tetravalent vaccine made by Takeda is going to have efficacy results reported very soon. There was a press release earlier this week. We're hoping that it will look good. We're hoping that it won't have signals similar to Sanofi. I'm a little bit concerned about that vaccine because in early studies, when you looked at viremia, it only has dengue 2 viremia. It doesn't have viremia to any of the other serotypes, and I'm a little bit concerned they may have a cross-reactive antibody problem as well. I will say if that vaccine fails, then I think we're in big trouble. I don't know how we're going to get a vaccine across the finish line. Yeah.
Yeah, thanks, Anna. In the Philippines, they did use Denvaxia, and I understand that it did, when it was withdrawn, or at least the plans were changed, I guess that sort of made a lot of headlines also. Do you know if there's any measure of, does this have an impact overall on vaccine hesitancy for other vaccines?
Absolutely. In the Philippines, vaccine hesitancy became quite profound, and vaccination rates for the other vaccines went down precipitously as a direct result of Denvaxia in the Philippines. And they're just starting to recover. It had a huge impact.
There's a big measles outbreak that some people have attributed to.
Absolutely. Absolutely. Yeah, it had a huge impact.
Thank you, Anne. What is the current thinking of the T-cell immunity side?
Okay. That also has been a paradigm shift. So early on, we thought that more severe disease with secondary dengue infection may be due to cross-reactive T-cell responses leading to cytokine storm. More recent studies done particularly by Daniela Weisskopf in La Jolla and Eva Harris's group, we now think that actually the T-cell responses can be protective. And I will say one of the studies that we did early on in our vaccine studies, we looked at the T-cell responses following each monovalent vaccine. And if you get a monovalent dengue infection, your CD8 response is really targeted predominantly toward that particular serotype. It's more serotype specific. But when we gave all 4 vaccine viruses together in a tetravalent formulation, that response shifted and moved to conserved epitopes. So you got a much broader conserved response with when you gave all 4 serotypes together. And so we think now with sequential infection that probably is what's happening as well, that you can actually expand your cellular immune response repertoire. And I think that's why only 4 people had infectious virus recovered. I think it's my hypothesis. We still have to do the studies to look, but I think it's the cellular immune response that's containing that virus before we can detect infectious viremia. Yeah.
Great. Well, thank you so much, Anna. Thank you for a great lecture. Thank you for the incredible science you do. And it's a privilege to have you on our faculty here in the School of Public Health. Thank you.